Hello everyone, welcome back to Dr. K. PREM-PRIMER video lecture series presenting by Dr. K. Prem, that's me. In today's lecture, we'll talk about the linker molecules, how they are useful in addition of cohesive ends to the blunt end molecules and their properties. As you all know that when you want to clone any DNA fragment into any vector, that DNA fragment need to have compatible sticky ends. Compatible sticky ends. You see, when you wish to clone any DNA fragment, compatible sticky ends are desirable on insert and vectors. So when you have a vector and insert with the same sticky ends, then the ligation process is very easy. Ligation process is easy. And also, the sticky ends are usually generated on vector and insert by the digestion with the same restriction endonucleases. So if the insert and vectors are digested by the same restriction endonucleases, then compatible sticky ends are generated and they are useful in the ligation of two different molecules. But when you want to clone a DNA fragment with a blunt end, there is no cohesive or sticky end, then what do we have to do? Then we have to add sticky ends to those molecules, especially cDNA molecules which are prepared from mRNA. They don't have sticky ends. They don't have sticky ends. Certain DNA fragments also have blunt ends. If that is the case, if you add sticky ends to them, then they are compatible to clone into any vector. But to add a sticky ends to the blunt end DNA molecules or cDNA molecule, there are three methods are available. One is linkers, and second one is adapters, and third one is a homopolymer tailing. So linkers are the chemically synthesized short DNA, double-strand DNA molecules. They can add it to they can link it to the blunt end DNA molecules and followed by the digestion with the enzyme, and it will generate the sticky ends. Adapters are the preformed cohesive ends are there, and those are attached to the DNA molecule. Then sticky ends are formed. Those are useful in the ligation. And the homopolymer tailing means here terminal deoxynucleotide transferase has a property to add nucleotides at 3' end. So if you provide A or T or G or C to the insert and a compatible nucleotide is added to the vector, then they are ligated. So today we'll talk about the linkers. So linkers, adapters, and homopolymer tailings are the joining molecules of different inserts and vector. They are the joining molecule. So we will see the linkers in today's lecture. You see, this is a typical structure of a linker. You can see this is a typical structure. Means it's a chemically synthesized double-stranded DNA molecule, double-stranded DNA molecule. It's an oligonucleotide. And also, and more importantly, it has internal restriction site. It's a double-stranded oligonucleotide with internal restriction site or restriction cleavage site and blunt ends. You see here, this is a typical linker where you can see the, it's almost 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and the 14th one. It's an oligonucleotide, and it is a double strand. It is a double strand oligonucleotide, and the sequence is known, and it has internal restriction site or internal restriction cleavage site. You can see here, it is having a GGATCC. This is a restriction site and it is a blunt-ended. Linkers are defined as chemically synthesized, double-stranded oligonucleotides with internal sites and blunt-ended. They are chemical, they are synthetic double-stranded DNA oligonucleotides, have internal restriction site and blunt-ended. Right? These are the linker molecules. The linker molecules have 5' phosphates and 3' OH like a natural DNA. They're having the 5' phosphates and 3' OH like a natural DNA. More importantly, the ends of the linkers are having the G or C, so that the linkage will be strong enough, right? And these are the linker molecules. You can see some of the properties of linkers. You can see, as I defined, linkers are chemically synthesized double-stranded DNA oligonucleotides with an internal restriction cleavage site and the blunt-ended. Second property, you can see linkers can be put onto blunt-end DNA fragments and cDNA molecules to generate a cohesive ends after digestion with the particular restriction endonuclease. So these linker molecules can be added to the blunt-end DNA molecules or cDNA molecule by ligation by employing the T4-DNA ligase. Once the linkers are added to the blunt-end DNA molecules or cDNA molecules, they can be subjected to the digestion and generate the cohesive ends. As I told you that linkers have 5' phosphates and 3' OH like a natural DNA. You see, ligation of linkers to the blunt-end DNA molecules is quite easy because you can provide the larger quantities of linker molecules so that the proportion of insert to the linker to the blunt-end molecule is so high so that the ligation happens easily. The same thing. Ligation of linkers with a DNA fragment is quite easy by providing an excess concentration of chemically synthesized linkers, even though it is a blunt-end ligation. The linkers are added to the blunt-end DNA molecule, right? So here, the ligation process between the linker and the blunt-end molecule is easy because you can provide the higher concentration of linker. Yeah, you see, during the ligation, linkers can bind to each other, and also they can bind to the linker, which has bound to the blunt-end DNA molecule and forms a chain-like structure. So linkers can form bind with another linker. Or it can also bind to the molecule. Linker can bind to the linker which is attached to the blunt end and form a chain-like structure. Chain-like a structure. We'll see that in the next slide. But here, whenever a blunt end molecule have certain restriction sites, so for example, it is have a BAMH1 restriction site internally. The blunt-end DNA molecule. We cannot use a linker which is consist of a BAMH1 because after ligation of linker with the BAMH1 restriction site to the blunt-end molecule, when you subject to the restriction digestion, the enzyme create cohesive ends in the linker as well as the DNA blunt-end DNA molecule. So when you are selecting the linker molecule, you have to choose whatever is not there in the whatever restriction rate which is not present in the blunt-end DNA molecule. Such linker you have to choose that such linker should be compatible to the vector. So in a blunt-end DNA molecule have EcoR1 site inside. If that is the case, then you cannot use the linker molecule with the eco-R1 site. If you use such linker, what will happen? Linker is having eco-R1 and your blunt end also have a eco-R1. When you subject to digestion, linker also undergoes the digestion and your blunt end DNA molecules also undergoes the digestion. So you have to avoid such situation, right? So you will see the here link. What is a linker? How you can add in a pictorial form. As I told you, this is a linker where it is a chemical synthesized double stranded oligonucleotide. That's a DNA, and it is having the internal site. You can see here you have an internal site GGA-TCC. That's nothing but a BAMH1 restriction site, and it's a 14 mark. You see, I have represented this is as a linker. BAMH1 is there inside. And also the linkers have 3' OH and 5' phosphate like a normal natural DNA. So you have to select the linker with the desirable restriction site. Here we have selected this linker with the BAMH1 restriction site because BAMH1 is not present in our blunt end or cDNA molecule. So you select the linker with the internal restriction site, that's a BAMH1. Then you take the DNA molecule, that's a blunt ended. It could be a DNA fragment or it can be a cDNA molecule which have blunt ends. So when you mix the linkers with a higher concentration to the blunt end DNA molecules and you keep for ligation. So you can see the linkers and the blunt end DNA molecules in a ligation mixture where you use the T4 DNA ligase to ligate the linker to the blunt end DNA molecule. So linkers plus blunt end DNA fragments and the T4 DNA ligase. So once the ligation happens, the blunt end linkers can attach to the termini of blunt end DNA molecules or blunt end DNA fragments or cDNA molecules. You can see the same thing. So after ligation, the linkers are attached or ligated at the termini of blunt end DNA molecules. See, both sides or both termini of blunt end DNA fragments are ligated to linkers. You see, you get a linkers ligated DNA molecule. Right? You will see linker plus blunt end DNA fragments, and that ligation, you get a DNA fragments with the linkers, blunt end DNA molecules with the linkers. Now such molecule is subjected to the restriction digestion. So here, see, you all know, we know that the linker is having the BAMH1 restriction site. So we use the BAMH1 restriction endonuclease to cleave the restriction site in the linker and to form a cohesive ends. So after digestion with the BAMH1 endonuclease, the BAMH1 endonuclease acts on the BAMH1 site in the linker and form the cohesive ends. You see, DNA molecule with the cohesive end. Now this DNA molecule with the cohesive end can be ligated with the vector with the same cohesive sites, same cohesive ends. So vector also having the BAMH1 cohesive ends, and insert also have the BAMH1 cohesive ends. Then go for ligation with the T4 DNA ligase, you get a recombinant molecule. So the aim of this experiment is to clone the blunt end DNA molecule with the help of linkers. Here you see, when a linker is attached to the blunt end DNA fragments, that's a blunt end ligation. Once the blunt-ended DNA molecules are ligated with the linkers, then you get a DNA molecule with the cohesive sites, cohesive ends, and those fragments are ligated with the vector, and you get a recombinant molecule. So I'm saying here, linkers are useful in the cloning of blunt-ended DNA fragments or cDNA molecules. So they'll once they attach the blunt-end DNA molecules, then digestion, you get a cohesive ends. Same procedure we'll see in the next slide. You see, first thing is linkers with the desirable restriction sites are selected. Then such linkers and blunt-end DNA fragments or cDNA molecules to be cloned are ligated. So once you select the linkers and this blunt-end DNA molecules are ligated. And they are ligated by the T4 DNA ligase, and ligated molecule with the linkers are subjected to the restriction digestion, right? You see, linkers are ligated to both terminal of DNA fragments, and linker and insert are ligated by the blunted ligation, and the DNA fragments with the linkers are subjected to the restriction digestion with the specific enzyme, which generates the cohesive ends. Then the resulted cohesive ends containing vector and DNA fragments are ligation by the 2,4 DNA ligase, and the cohesive end ligation happen between the vector and insert. Then finally you get a that ligation picture is transformed to screen the and select the recombinants. So at the end, I'm telling you all that linkers are molecules which have a restriction site internally that can be added to the blunted DNA molecules. Once the blunted DNA molecules with the linkers are digested with the restriction nucleates to generate the cohesive ends, those the DNA fragment with the cohesive ends can be ligated with the vectors with the compatible sticky ends to form a recombinant molecules. So this is all about linkers. So to get more update from my channel, try to subscribe. That is Dr. K PREM-PRIMER. If you have any issues, like, if you have any doubts, write to me and I'll get back to you. And if you like it, try to give thumbs up. If you want to share with your friends, there is no issue, try to share. Thank you all for listening. See you again. Until then, bye-bye.